So now we're going to talk about how viruses protect themselves against adaptive immunity. So adaptive immunity usually occurs later than innate immunity. It is a more specific and targeted response rather than innate immunity, which basically targets everything, and it takes a longer amount of time to induce as well. But obviously, like, viruses have evolved different strategies to evade this adaptive immunity of ours, which is why like they have been so successful. And in particular, it is the DNA viruses which have developed some very interesting strategies against adaptive immunity. And just to refresh, the DNA viruses include parvovirus, adenovirus, papovavirus, including HPV, hepadnavirus, which includes hepatitis B, herpes, and pox, which includes vaccinia. And yeah, it is mainly the DNA viruses which have adapted so many strategies because its large genome allows it to have host homologs of genes, and these homologs can act in the place of host genes, thus inactivating them and preventing host genes from doing their jobs, or host proteins. Okay, now we're going to talk about strategy number one. Strategy number one is latency. So it enters into a non-replicating phase where it doesn't produce much structural proteins to be detected by the immune system or the adaptive immune system. And if it doesn't get detected, obviously it doesn't get killed. And then it protects its genome during this case, and then when the immune system is weak again, you might strike and enter into its vicious or infective form again. And examples of these include herpes simplex virus, varicella zoster virus, EBV, which are all part of the herpes family, and HIV, which is a retroviridae, but it's kind of special because it belongs to Baltimore class 7 and is able to reverse transcribe its DNA. Its ssRNA, sorry. The second strategy is just to exhaust the immune system, and then you can achieve this by using, in the case of hepadnavirus, was it hepadna? Yes, so HBV, it produces HBs virus-like particles, and these just stimulate the immune system to continue responding until cells die. And HCV as well, which I don't think is a DNA virus. So HCV is not a DNA virus, but it belongs to the flavivirus family. Maybe we'll just make a note. Flavi, hepadna, retro, papova. The next strategy used by papillomavirus is to cause, silent effects and not kill the cell. Then other things like measles, which is caused by rubiola, which is a negative ssRNA virus. So that causes SSLE, and the way it does this is by entering into a form of latency, I suppose. And then there is also antigenic variation, which is done by influenza and HIV. Influenza is part of the orthomyxo, negative single-strand RNA virus. HIV is retrovirus again. And basically, by changing its antigen and epitopes all the time, B cells can't recognize it. So this won't lead to aggregation of infected cells, immune clearance. It will stop Fc-mediated uptake into mononuclear cells, neutralization, recognition, all the good stuff. And what instead it allows the virus to do is to get endocytosed, uncoded. And what's interesting is that despite so many changes in their glycoproteins, these glycoproteins are still functional. Other strategies used by influenza include changing T cell epitopes as well. So we would know that the epitope that's presented by two T cells is derived from proteasome-directed degradation of the virus and presentation of the peptide on MHC to the T cell receptors, followed by activation. But what instead some viruses do is that they inhibit the maturation of dendritic cells, which are necessary for the digestion of the virus and presentation of the peptide MHC complex. So the HSV inhibits the activation of dendritic cells so that they can travel to the phagocytose the virus and then present the peptide MHC. Vaccinia virus also stops this signal transduction via the TLR4 because it resembles it. HCV and Vaccinia, they also inhibit the cytokine-induced maturation of dendritic cells, so they can't go on to present it. And then other strategies include evading the CD8 T cell responses. So we just evaded the CD4 responses and some CD8. And basically we can do this by downregulating MHC class 1 expression on the surface. HIV does this using a NEF protein, which endocytoses the MHC1 protein for CD8 recognition. It also does this by changing the antigen so that the peptide epitope can't be recognized by the T cell receptor. Cytomegalovirus uses protein US6 to bind to the cytosolic side of the TAP transporter necessary for transporting the MHC peptide complex from the endoplasmic reticulum to the cell membrane. And by doing this, no T cell recognition. HSV ICP does the same thing, but it binds the cytosolic side. It binds to the cytosolic side of the cytosolic transporter. Of the TAP transporter, sorry. Yep, so this is unnecessary over here. Okay. So there is no translocation of MHC class 1 across the ER secretory pathway. Adenovirus does pretty much the same thing. It binds to the MHC peptide complex and retains it in the ER, and there is no TAP recruitment for getting that out of the cell. Ebna virus in B cells prevents proteins from being degraded by the proteasome, so there is no peptide to be presented. And HIV, TAT, RSV, adenovirus all decrease MHC1 expression. And mouse norovirus in shutting down host cell translation does that to MHC class 1 as well. So obviously if you are hiding from the T cells, you might still get attacked by the NK cells. So you need strategies to evade this. So although the CMV cytomegalovirus has the protein US6, which allows the retention of the MHC1 in the endoplasmic reticulum, so there is no CD8 T cell recognition. It has other ways of stopping the natural killer cell from killing it as well, and these include retaining the activation receptor for natural killer G2D cell receptor in endoplasmic reticulum, so that NK cells can't be activated. This is for the mouse, and this is done using the protein M152. For humans, you might upregulate the expression of HLA-E, which bind and present peptides of other MHC. You might also express an MHC1-like molecule UL18 on the infected cells, which bind and activates the inhibitory natural killer cell receptor, which inhibits killing. Lastly, you have some interfering strategies for cytokine production, and these could vary between interfering with the synthesis, to the function, to the response to cytokines. And vaccinia and myxoma, they both bind to cytokine. They both have interferon receptors homologs which bind cytokines and cause inhibition. EBV has an IL-10 homolog which inhibits the Th1 response. POX inhibits the cleavage of IL-1B precursor, which is necessary in apoptosis. Adenovirus inhibits TNF-alpha mediated killing, inflammation, apoptosis, etc. POX virus inhibits the ILB1 cleavage, which prevents the formation or activation of caspases. Lastly, they might exploit autophagy as a means of replication. So usually you would get autophagy of damaged organelles, and then you get a decrease in pH and lysosomes, which kills everything that is within the autophagosome. But some viruses can actually exploit this machinery to use for their own replication, and then this isn't detected by host cells as well because it's enveloped. And lastly, you can get some complement evasion as well, but obviously this only applies for the enveloped viruses which are big enough to be attacked by the complement proteins, such as the pox family of viruses, including the vaccinia. And what this essentially has is something to bind complement 3B and 4B. And that's it. Thank you so much for listening. Have a good day.